Hello and welcome back to another lesson. So today we're going to be talking about meiosis. And meiosis, basically, it's going to be a continuation of our last lesson on in biology, which was mitosis. And you're going to see a lot of the similarities between the two when we talk about them. So let's get into it. So just to give you a little bit of breakdown of meiosis produces gametes, which are sex cells, and each cell that is produced has half the number of chromosomes than the other cells in the body, so it's called haploid. Now, for example, humans have a diploid nucleus, meaning that the nucleus contains two copies of each chromosome in a homologous pair. So you have 46 chromosomes in 23 pairs. Sex cells have a haploid number because they fuse together during sexual reproduction, and that's done so to keep the same number of chromosomes. Because you're fusing and you want to keep the same number of chromosomes, initial gametes, or the cells that are involved in that process, need to be half. And in meiosis, the actual diploid number of chromosomes, which of the parent cell, is actually reduced to a haploid number. And again, like I said just before, that it's fused back into a diploid number during sexual reproduction. Now, for the phases, you can see that the phases are very similar to each other, and they're actually very similar to mitosis as well. So the first phase of meiosis is prophase 1. And prophase 1, it's the same thing as in mitosis, where you have the nuclear envelope breaking down, but there's one big difference in that there's crossing over of the chromatids, meaning that you're basically shuffling up the genetic deck. And you're doing this because that's what meiosis is. You want to have that genetic variation, which you don't have in mitosis, because mitosis is you want to create the same cells, and you're doing this because you're repairing cells, or you want to replace cells. So you want to have that genetic. You want the cells that are created to be genetically identical. But in meiosis, you're making gametes, and you're making sex cells which pass on the genetic identity of the individual. So you want to have a little bit of a genetic variance, otherwise you would look the same as your parents, which you don't, obviously. So this is where the crossing over and the shuffling of the genetic deck happens in prophase 1. Now the next phase is metaphase 1, again similar to mitosis, but this time you're actually lining up the homologous pairs rather than the actual chromosomes. So, for example, let's say in this cell you have 4 chromosomes and 2 homologous pairs. What's happening is the actual homologous pairs line up, and the spindle fibers come and attach themselves to it. And you'll see that in anaphase 1, the homologous pairs are the ones that are pulled away. So you still have, when you make, and then you go to telophase 1, and when you have these two, that shouldn't say diploid, you have these two daughter cells. These two daughter cells, which have the two homologous pairs. So you have the 4 cells that were in the one cell before are now 2 cells, or 2 chromosomes. Sorry, the 4 chromosomes which were in the one cell are now 2 chromosomes which are in 2 different cells, and in telophase you have the cytokinesis, which removes the 2 cells. But the process isn't done there. Next you go on to prophase 2, and this is the same as prophase 1, except there's no crossing over. There's no crossing over. And this is going to be similar to prophase in mitosis. There's no crossing over. It's just the breaking down of the the nuclear envelope. And metaphase 2. Again, this is all the second phases, or the second section, is basically the same thing as what happens in mitosis, essentially. And in metaphase 2, the chromosomes are the ones that are lining up, whereas in metaphase 1, it was the homologous pairs, but now you're having the chromosomes line up. Now, when you go to anaphase 2, you have the chromosomes which are pulled apart. And again, recall, Then anaphase 1, it was the homologous pairs which are pulled apart. And the last phase, telophase, you end up with 4 haploid cells, which are all genetically different and have half the number of chromosomes which the original parent had. So again, let's talk a little bit about the difference between mitosis and meiosis. Again, like I said before, mitosis is used to repair and replace cells, while meiosis is used for the gamete formation. Mitosis produces 2 daughter cells, 2 diploid daughter cells, which are genetically identical, where meiosis produces 4 haploid daughter cells, which are genetically different. So those are the two main differences between the two processes. Hopefully you learned something today, and tune in next week, or come back again for another lesson.